next question is, the pH of soil influences the availability of ions as well. So what do you think is the change? So let's say magnesium and potassium, then we have nitrate and phosphate, and iron and sulfate. So the pH
Okay.
scale is given below, where the activity can be maximum or lowest. Name the ion that is least available in soils at pH 4. Let's move to pH 4, which has the least values.
Potassium and
Potassium,
sulfur.
potassium and sulfate. Okay. Potassium and sulfate would have the
Least.
lowest one. Okay. So if we compare potassium and
It could
sulfate,
also be magnesium actually,
okay.
like at
Okay,
4.
so we have this curve, we have this curve, and we have this curve. Clear? Now, which is
Yep.
showing the least activity, which is not increasing at all?
Oh, okay. That's potassium.
That would be potassium. And why haven't you selected phosphate? Look, my question is still there. Why haven't you selected phosphate?
Because,
It's closing.
yeah, it's
It's a
closing.
zero. So this should be our like main
Okay.
priority, right? So
Okay.
at pH 4, phosphate is showing the least available. Okay. What is least at pH 11? The last point. Look at this peak,
I think that's iron.
iron. So we have to look at the like least value in which the peaks are joining, or they have some uh, connection point.
Okay, okay.
Okay. Our next question is, plants grown in soil of pH 10 may show symptoms of deficiency. Let's say at 10 we have less this available, we have less this available. So
Yeah.
what could be the deficiency of magnesium ion, and what could be the deficiency of nitrate ions? That we have discussed already.
Yes. Magnesium would like cause chlorosis, like if there is less of it.
Okay, then you need to explain chlorosis. What is the meaning of chlorosis?
Basically when like the chlorophyll isn't green anymore, like the pigment is lost, like it becomes like the plant becomes sort of yellow, like the leaves.
The plant becomes yellow. Okay, because of the unavailability of chlorophyll. Okay, what is the deficiency of nitrate?
Um,
It's related to proteins.
yes, for
Okay,
like growth and development.
so there would be less growth, there would be less development.
Yes.
What else can be
Oh,
written?
also enzymes.
Also, the
Enzymes.
enzymes are not working.
Yeah.
Okay, correct. Let's move to the next question. Okay, again, an experiment is given, and we need to go through the experiment. Okay, they have used certain things. They have used the lamp, the stop clock, movement of, they have used the scale, and then there is an air bubble, there is a syringe, there is like a test tube, pond plant, and a ruler. So
Okay.
what are the uses of the instrument that have been used? So let's say our first
Um,
instrument is the stop clock. What is the use of this instrument?
um, to like measure the amount. Wait. I was going to say like
Okay, okay. Let's go through the question first. Then we'll discuss this. A
okay.
student set up an apparatus as shown in this figure, and they are investigating the effect of light intensity. That's why they have using a scale, a ruler,
Oh,
so
okay,
that it
okay.
can be moved to change the intensity. Okay. The student maintain a temperature, so there must be thermometer as well, and measure the distance traveled by the air bubble. And here we again have a scale in which we are determining how much this bubble is moved.
Okay, so they measure
Okay,
after like each 5 minutes.
each 5 minutes. So why we are using the stop clock?
To like measure the time.
To measure the time. Correct. Okay. Why are we using this gas syringe?
Um, to like insert the.
To do what?
Are we trying to see like how many gas bubbles are collected?
Okay, how many gas bubbles are collected? What else can be? So syringe is basically used to collect gases.
Yeah.
But here we have to check the movement of this air bubble.
Okay.
Okay. So if oxygen is producing, it's going to collect somewhere, and it's collecting over here. They're mentioned
Okay.
over here.
Okay.
The gas syringe is used so that the it's not going to move out or somewhere else. It will stay in the system.
Oh, okay.
Okay. If it stays in the system, it means it won't be disturbed. So it would.
But couldn't they have like just closed it somehow? Why did they have to use the syringe specifically?
Okay. So when the bubble is produced or gas is produced, this is going to move. If there is a space over here, it's maybe going over here, or maybe not move due to the presence of what pressure over here, or the
Oh,
collection of
okay.
gas over here. It won't move. Maybe.
Okay.
So we are giving it space, or we are basically helping the bubble to move through it. Or the correct word that we have to use is repositioning of the air bubble.
All right.
Okay. Okay. Why do we use this glass tank with water? Now we have placed a glass tank between a lamp and a test tube. Why do you think so?
To like um lower the light intensity.
Uh, okay. Think like you are correct. It lowers something, but not the light intensity.
The temperature?
The temperature, because every light
The
is
temperature is
has
like
produced some
increasing.
heat. So this glass tank is absorbing that heat of light.
Yeah.
So it's maintaining the temperature. It acts as a water bath, you can say. It's maintaining a temperature. Okay. So our
Yeah.
question is, why do we use glass tank and why do we use syringe?
Yeah.
So we
So
use,
glass tank we use to maintain the temperature.
and syringe is used to
To keep like the air
repositioning
bubbles.
of the air bubble.
Yeah.
Okay. So explain why the air bubble moves down the capillary tube. Okay. Why this air bubble has moved here? There is collection of gases at the top.
Yes.
So water level will decrease. So it means the bubble is going to move down.
Okay.
Now tell me what's going to happen. Plant, due to photosynthesis, plant produce oxygen.
Yeah. And when oxygen is released from the like the top of the plant,
Okay.
since the water potential at the top of the plant is lowered,
Again,
the
no, no,
what?
no. We don't have to talk about the water potential here, because water potential is
Oh.
related to salt concentration.
Okay.
So we won't mention water potential over here. We'll
All
talk
right.
about the collection of oxygen at the top due to photosynthesis,
Yes.
and it pushes the bubble down the capillary tube.
Hmm.
Okay. It's a concept of chemistry as well. It's
Okay.
about collection of gases. When
Hmm.
gases are collected at the top, because oxygen is denser,
Yes.
then water is collected on the top. Now water level will decrease. Okay, and when
Okay.
water level will decrease, it is going to move the bubble down. Water is moving down. Water is collected here in this beaker, let's say,
Hmm.
when it's moving down. Okay.
All right. All right. Yeah.
So it's a pressure that is exerted by the gas which is collected on the top, which moves the bubble downwards.
Okay.
Okay, let's move to the next question.